Okay, in this video we're going to talk about active membrane transport. Remember, with active transport, this is one that requires ATP to move substances or molecules across the membrane. And there's basically two major types of active membrane transport. We just have active transport in general, and we'll differentiate between the subtypes of this. And vesicular transport, which is still considered active membrane transport because it requires ATP to move vesicles across the membrane and within the cell. So both these require ATP. And the reasons why you might need additional energy to move a substance is that it could be that the solute's too large for channels, so it can't fit through a particular type of channel, so it may require additional energy to help to transport that larger molecule. Or if the solute's not lipid soluble, so it can't just diffuse across the membrane, or the solute is not able to move down the concentration gradient. For instance, if you're trying to move a molecule or some sort of solute from low to high concentration up the concentration gradient, that's going to require some additional input of energy in the form of ATP because you're basically going against the tendency of nature. And that's going to be like moving up a hill from low to high point or from low concentration to high concentration. So this is when it would require active transport. So with active transport, we can use solute pumps or carrier proteins, and these are the ones that bind specifically and reversibly with a substance that's being moved. We've talked about carrier proteins already with a facilitated diffusion channel. However, this is an example of carriers that actually, they work similarly, but they require the use of ATP to pump a molecule in a particular direction. Now we have two types of carriers. We've got antiporters and symporters. An antiporter is one which transports a substance into a cell while transporting a different substance out of a cell. So anti, meaning against, is a type of carrier protein that can actually help transport molecules in opposite directions. Symporters are one which transport different substances in the same direction. So an example of a symporter could be like sodium-glucose transport, where sodium and glucose move in the same direction into the cell. And because they're going in the same direction, we call that a symporter. Now, what we find then is that for active transport, we're moving solutes against their concentration gradient, right, from low to high. And because you're going from low concentration to high concentration, you can't rely on diffusion. Remember, diffusion goes the opposite way. Diffusion would have a tendency then for molecules to go from high to low. But if we're trying to move molecules against diffusion, that's going to require some additional energy in the form of ATP. Now there's two major types of active transport. We got primary active transport and secondary active transport. Primary active transport is one which requires energy that comes directly from ATP hydrolysis. That's why it's primary, because you primarily use ATP in the most direct way to basically help transport or transfer that chemical energy into more of a kinetic type of energy, and that way you can move a molecule from one area to another. Now, if you're using ATP directly, we call that primary active transport. Now, secondary active transport is actually where you require energy obtained indirectly from ionic gradients. So I'll explain this one further when we get to a picture here. But secondary active transport relies on the stored potential energy in an ionic gradient across the membrane that came from other primary active transporters. But I'll explain this one here further. Now, primary active transport, remember, is energy from direct ATP hydrolysis, causes a change in the shape of a protein, and that causes the solutes or ions that can be pumped across the membrane against their diffusion gradient. So examples of different types of primary active transport pumps we have in a typical cell would be things like calcium transporters, hydrogen transporters or pumps, and also the sodium-potassium pump, which helps to pump sodium out of the cell and potassium in the cell against their diffusion gradients. And that's why it requires ATP hydrolysis. Now, the sodium-potassium pump is one of the most studied pumps because basically every cell in your body has a sodium-potassium pump. It's an enzyme, and actually we also call it the sodium-potassium ATPase because it's an enzyme that uses ATP to pump sodium and potassium. Now, it pumps sodium out of the cell and potassium back in, and it's located in all plasma membranes, but especially in excitable cells like nerves and muscles. Now, sodium-potassium pumps work as an antiporter because it pumps sodium out of the cell while pumping potassium into the cell, and because those are moving in opposite physical directions, we call that an antiporter. And it requires additional energy because you're pumping those ions against their concentration gradient. So sodium is already in higher concentration out of the cell, but we're using ATP to pump it in that direction. Now, it maintains electrical chemical gradients. That's the whole purpose of the sodium-potassium pump. And this helps to separate certain electrical charges across the cell and also maintains appropriate water balance for your cells. So this is just showing what the sodium-potassium pump is doing. So we want to pick a starting point here. We'll pick this part. So what we find then is that here's our plasma membrane. We have our sodium-potassium ATPase here. This is the extracellular fluid and the intracellular fluid. You see that normally in the extracellular fluid, sodium is already in high concentration. And normally in the intracellular fluid or cytoplasm, you find that potassium is in high concentration. And the way that we maintain these gradients is using this sodium-potassium pump. We can actually use the chemical energy of ATP, remember the stored chemical potential energy to ATP, to help physically move sodium out of the cell. So sodium atoms bind here, and once they bind, the transporter changes shape and helps to move that sodium out of the cell and into the extracellular fluid. Then what happens is some potassium atoms come in here, or ions, and they bind to their binding sites within the enzyme, and then this enzyme can rock back, and potassium then flows into the cell. Now this rocking motion of the sodium-potassium pump, back and forth, requires the use of ATP, and in that regard too, you actually can move these atoms against their concentration gradient, right? So moving sodium to an area of higher concentration is against diffusion, so that requires ATP. And then also moving potassium to an area of higher concentration, which is also against its concentration gradient, so that you need to also use ATP there. However, you're only using one ATP molecule to pump 3 sodiums out for every 2 potassiums in. So it's fairly efficient. So secondary active transport depends on these ionic gradients. So let's say for the sake of argument here that the sodium-potassium pump is what maintains the gradient of sodium and potassium across the plasma membrane. So the reason why sodium is low inside of a typical cell is that, well, it was pumped out. And the reason why potassium is in high concentration within a particular cell, well, it was pumped in. Now because there's a separation in concentration, we call it a gradient, but that requires the use of energy to even create that gradient. So that the energy that's stored in these gradients can be used to transport other molecules. So for instance, let's say that you have low sodium concentration that's maintained inside the cell by the sodium-potassium pump, and it strengthens sodium's drive to want to flow into the cell. Now what's interesting here is that sodium can physically drag other molecules with it, but this is through a different type of transporter, usually a symporter of some sort. So things like sugars, amino acids, and other ions can be transported into the cell through secondary active transport, because if you allow sodium to flow down its concentration gradient back into the cell, it can bring another molecule with it. And that's why it's secondary active transport, is that you're not using ATP directly, rather you're relying on the concentration gradient that already exists across the cell, and that's stored potential energy there, chemical potential energy, and you can use that energy to help transport other molecules like sugars, amino acids, and ions. So looking at the example here, remember primary active transport could be like the sodium-potassium pump, where you use the chemical energy of ATP to pump sodium out and 2 potassiums back in. Now let's say for second active transport, what if we wanted to get some glucose into the cell, but glucose is already in high concentration here. So we can't just allow glucose to move through a facilitated diffusion channel because you're trying to bring it from low to high concentration, which isn't going to work because that goes against diffusion. So what we need then is for glucose to kind of piggyback on the movement of sodium here, where sodium does want to flow into the cell because sodium's in low concentration here, so that the movement of sodium down its concentration gradient can piggyback glucose with it to help bring glucose up its concentration gradient. And that's an example of secondary active transport. Remember, so the diffusion of sodium down its concentration gradient can kind of draw another molecule up its concentration gradient with it. And because you're relying on the potential energy in a concentration gradient, that's why we call it secondary active transport. Now, vesicular transport involves the transport of large particles like larger macromolecules and even fluid across the membrane. And we use membranous sacs called vesicles. So basically it's a, kind of a lipid bilayer like the plasma membrane, and there's typically a fluid-filled center, and this is what we call a vesicle. Now the movement of these vesicles usually requires cellular energy, and that's typically ATP. So in terms of vesicular transport, we got several different types. We have endocytosis versus exocytosis. Remember, endo means within, so endocytosis means transport into the cell. And there's 3 different types here: phago, pino, and receptor mediated endocytosis. And then exo, meaning out of, exocytosis is actually a transport of vesicles out of the cell. And then trans means across, so transcytosis means transport into, across, and then out of the cell. And vesicular trafficking is just basically where you have a vesicle that's moved from one area of the cell to another. So in the following slides, we're going to differentiate between these different types of transport. So what we see here is basically, kind of a mixture of all those different types we saw there. So if you're saying that you're bringing a vesicle into the cell by basically having the membrane fold inward and then pinching together to form a vesicle, this is an example of endocytosis. Now, this endocytic vesicle can be used for a variety of things. Like you can actually, help to fuse this with an endosome or a lysosome to help break down different molecules inside that vesicle. Like if these are little bacterial cells, you can take a lysosome and fuse it with this vesicle to break down those bacteria. You can actually digest these and transport that back away from the cell. Or you can just actually have endocytosis here, transport across the cell, and then exocytosis on the other side. And so if you have a mixture of endo and vesicular trafficking and exocytosis, we actually call that transcytosis, going this way. Now, phagocytosis, phago means to eat. So phagocytosis is when the cell engulfs a large particle, and it does this by projecting these pseudopods or false feet around the particle. Now they enclose that within a membranous sac called a phagosome, and this phagosome is usually combined with a lysosome to kind of break down this molecule. Like this might be some waste or debris or even a bacterial cell that you might want to digest. Now undigested contents remain in the vesicle, and we might call that a residual body that can be ejected by exocytosis. Now pinocytosis, well, pino means water, and pinocytosis is when the cell takes large gulps of extracellular fluid. Now this can contain some solutes, and there's no receptors used here. The process is nonspecific, and these vesicles are protein coated, but it's just a way for the cell to bring in extracellular material, into the cell, and maybe even bring that across the cell as well. Now there's receptor mediated endocytosis, and this is basically endocytosis that's triggered when a molecule binds to a particular receptor. So an example here could be something like insulin binding to its receptor can cause the membrane here to have endocytosis of the receptor hormone complex. And then what happens then is that once you have this vesicle inside the cell, enzymes can be used to digest or kind of cut away the molecule from these receptors, and you can bring this back towards the membrane to recycle those receptors. But this is a pretty common activity that cells can use to recycle their receptors. Now, what we see here then is exocytosis, and this is a process where material is ejected from the cell. It's usually from cell surface signals or changes in membrane voltage, and a substance being ejected is enclosed in a secondary vesicle, and the protein on this vesicle called V-snare finds a hook called T-snare. They actually interlock through a process of docking, and that can trigger exocytosis where the vesicle fuses with the membrane so that the fluid center of that vesicle is dumped out of the cell, or exocytosed. So some different things that are exocytosed are basically transported out of cells are hormones, neurotransmitters, mucus, and even cellular wastes. So this is just showing that basically that process here where you have a vesicle here with your V-snare associated with it that can interlock with the T-snare here on the plasma membrane, and once they fuse and intertwine, the membranes here are allowed to fuse, and that way the fluid-filled center of that vesicle becomes continuous with the extracellular fluid, and those molecules are dumped out. Now, this docking process requires ATP, and that's why we still consider this active transport. And this slide here is just showing a photomicrograph or electron micrograph of of essentially exocytosis in a living cell. So you can see here's the vesicle, and it's fusing with the plasma membrane. You can see all those cellular contents just being dumped out. And it's basically the process of diffusion that allows these molecules to move from the vesicle into the extracellular fluid. And we're seeing this at 100,000 times visual resolution, so it's very, very tiny.